Gary mentioned my background in molecular biology. I worked in the biotech industry for 30 years and sold reagents to molecular biologists, mostly working in human and maybe some agricultural molecular biology. To my knowledge, no one's applying a lot of that to goldfish in particular, but some of the passive knowledge that I learned through my career, I got some insights, which I think I can be helpful in explaining some of the concepts. As well, I want just to kind of make sure people understand what genes are and what they aren't. Sometimes you hear people throw the word a gene for this, a gene for that, casually, not really understanding what a gene is. Like I'll hear the gene for a ranch, like what level does genetics control specific traits versus something as little as a protein or the expression of a protein. I don't want this to be too complicated, but as I go through talking about chromosomes and genes and alleles and loci and things like that, if something's not clear, just ask me. If it's too repetitive, just tell me to speed up and we'll move on. Okay, so preview, we're going to talk about chromosomes, DNA, genes and proteins, mutations, inbreeding, outcrossing, line breeding, and backcrossing, and some observations made by some goldfish researchers. These are some Japanese researchers. So you've probably heard the term chromosome before, and you have chromosome numbers. You hear about diseases caused by extra chromosomes or missing chromosomes. So chromosomes are bundled DNA. So if you think of DNA as the double helix, it's two strands of nucleic acids wrapped around each other, and then it gets tightly coiled and wound up in discrete packets inside the nucleus, so that when the cell divides, each chromosome can kind of reproduce itself and split in two. And when DNA is inherited, it's coming over as a whole chromosome, so that one chromosome may have multiple genes along it, but you can't easily separate the genes that are contained on a single chromosome because you either inherit the whole chromosome or you don't. And that's called linkage. You may hear about gene linkage. It's because they're close to each other on a chromosome. So there's an example. It's a graphic of what I just described with the double helix of DNA, and you can see how it gets coiled and packaged into these chromosomes, which are contained in the nucleus of the cell.
So when you breed the goldfish and the eggs are fertile, after about 8 hours, you can see polar bodies in the eggs themselves with your naked eye. Little black specks, right?
I've seen what you're talking about. I can't confirm that that's what they are. I don't know if a polar body could be seen with the naked eye. But I've seen, I don't know if it's like a little globule of fat. I would think a polar body would be an extremely small little thing. But I've seen, as a sign of fertilization, I do see these things. Yeah. I agree. But and I know you call them polar bodies, and perhaps they contain the polar body, or they are, but I can't independently confirm that that's what those are.
Everybody seen that? Yes. So when you get fertilized eggs, you got them in a bowl or on a mop or whatever. You can, they spawn in the morning, you put them in a bowl, and you're all happy, and you go to work, and then you come home, and your first question is, well, are they fertile? And after about 6-8 hours, you can look at the eggs, and most of the time, you can see inside these eggs with your naked eye these little black specks, and the hobbyists typically call them polar bodies that you can tell that the cells are dividing. So, sorry, Rob. Didn't mean to sidetrack you.
That's all right. So what a karyotype is something that they can do. They actually look under a microscope and spread out the chromosomes and take a picture of them during or cell division, and you can get the actual chromosome number. Now, chromosomes occur in pairs, so the chromosomes are numbered, but each one has one from each parent. And an important thing to realize about goldfish and some other species is that they're what are called tetraploids. So a normal chromosome contribution would be called diploid, where you'd have one from each parent. Well, somewhere during the evolution of the goldfish, they duplicated the entire genome and ended up with 4 chromosomes of each one. So a typical cyprinid, which is the family that goldfish belong in, have 26 chromosomes, but goldfish have somewhere, they're saying, between 50 and 52 chromosomes. And they say, well, how does that happen? And what's believed to be the mechanism is during meiosis, which is when the sperms and eggs are formed, the cell's supposed to split in two, and you get a haploid cell, which has half the number of chromosomes that's supposed to. And then when it marries up with an egg, say a sperm and an egg, it recovers the full diploid amount. So what could happen is during meiosis and division, if something goes wrong, all the chromosomes end in one cell and the other cell has nothing. So you end up with a diploid sperm or egg cell. And if it would be a rare event, but if that were to meet up with another diploid gamete, sperm or egg, you've doubled the number of chromosomes. Now this makes for a viable organism because it has an even number of chromosomes and has all the chromosomes. It shouldn't really cause the animal any problem because everything's expressed in the right proportions. You still have, everything expressed the same. And over long periods of time, we're talking thousands, millions of years, these chromosomes will inherit mutations along the way and slowly change so that they're no longer 4 identical, but start moving back toward 2 different separate pairs instead of 4 identical, 2 separate pairs. So you're going from being a tetraploid back toward a diploid because now of the 4, 2 are starting to behave like a pair rather than an identical quartet. And then they start accumulating new mutations. So it's just the whole process of evolution, how that works. And it accounts for why you see such variation in goldfish. I mean, you don't normally see such plasticity in other animals where you can have dorsalists survive and then double tail and big eyes and things like that. And the reason is when these mutations occur in a diploid animal, they're often lethal. I mean, the embryo will just stop developing. Whereas if this mutation occurs in one of 4 chromosomes in a goldfish, it will affect the expression. You get this new phenotype, but because there's 3 other copies of that chromosome, you still have enough of the normal protein or gene being expressed that it's no longer lethal. So it allows you to have living, mutants that come out, which makes goldfish very interesting.
Does it happen in any other species?
Yeah, a lot of, and it depends on how long ago it happened. It's a mechanism for speciation. I mean, new species are slowly coming to, and they found lots of Corridor's catfish are tetraploids. It tends to happen more commonly in cold-blooded species, where they're producing millions and millions of eggs and gametes that are subjected to the environment where you have, pressure and things that affect cell division. So the odds of this rare event happening in the ocean, with millions and millions of fish eggs and a massive spawn, even though it's a rare event, it does happen. It's much less frequent in mammals, and they can make triploids, which are 3 chromosomes, artificially by using pressure on eggs during their development. And you've probably heard of triploid grass carp, which are sterile. And the reason is grass carp are great for clearing out weeds in ponds, but if they get out into the wild, you know, what's happened like in the Great Lakes and places where the different carp have been introduced, they wreak havoc. So they do have techniques for producing sterile triploids, but then again, once you get back to an even number, like a tetraploid, this animal will be fertile again, and it will be a new species. These are the chromosome numbers, just for example, in common animals. You can see goldfish are way up there at 100 to 104 chromosomes. We said before was 50. That's the number of pairs. This is the number of, say, individual chromosomes.
So is that why there's so much variation in dog breeds too? Because they're fairly high as well? 78?
That's a good question. There is a lot of variation in dog breeds. A lot of that is probably due to the fact that they're one of the oldest domesticated animals. And I think as far as domestication, goldfish probably have only been kept for a couple thousand years or something, but dogs potentially go all the way back to the earliest times of human evolution.
Something special about dog genetics too, because they've been able to do it with foxes as well. Have you heard about the Russian foxes?
Absolutely.
The tails and the different coat colors?
Yeah, and there's something about domestication in general, and I've looked into this melanin, which controls a lot of things in coat color, and even in goldfish you have melanin expressed at different levels, which accounts for the different colors. Whenever you start domestic breeding of things, it messes up that melanin and you get the piebald patterns. You think about cows and pigs and horses, you don't see those broken patterns in wild animals as much unless they're very inbred. And I've seen small herds of white-tailed deer that are kind of trapped, and you do get some piebald ones coming out of there. Okay, so the normal process of cell division is called mitosis. This is what happens in what are called somatic cells. These are the cells in your skin and your tissues and everywhere, where you have a diploid cell. All the chromosomes line up in the middle. You have centromeres at the end. The chromosomes duplicate, and each daughter cell gets a full diploid number of chromosomes. The other process is called meiosis, which is gametogenesis, and this is how the egg cells and the sperm cells are formed. So you start with diploid cells in the testes and in the ovaries, and this is where polar bodies actually come from during the meiosis stage, where you have a primary oocyte, then goes to a secondary oocyte, and you get down to the haploid where it only has one of each chromosome. And during that process, it kicks out the other part of the chromosome. So only one of them becomes an egg. The other chromosome is just a little discarded piece. And that may be still contained within the mature egg as a little enclosed thing. I'm not certain if it stays there or if it stays somewhere in the ovary or is lost. A similar process in spermatogenesis. Moving on to what are genes, and this is a relatively recent, like within the last 30 years, discovery. It was long thought that genes, you had protein coding genes, and then there was long stretches of DNA that didn't code for a protein, so they called them the junk DNA. These were termed introns and exons. So the exons are the important ones that coded for proteins, and the introns were like spacers of random DNA nucleotides that didn't code for any known protein. They were thought to be unimportant. But what they've found out more recently is that it's not junk DNA, that mutations that occur in those intervening DNA sequences that don't code for proteins are important because they determine the rate of expression of the protein coding genes. So there are little sequences there that proteins bind to the DNA or RNAs bind to the DNA that triggers the RNA polymerase to make RNA off of that DNA template, and that would move forward into protein. And you've got to remember there are no genes for phenotype. And if you're not familiar with the word phenotype, phenotype is what something looks like. And genotype is its genetic type. So something can be either homozygous or heterozygous. I don't know if you've heard these terms before. So for example, I can think back to when I raised Dutch rabbits. You had black ones and blue ones. A homozygous Dutch rabbit had 2 of the black genes, one from each parent. You'd have all black offspring. A heterozygous one would have 1 gene for black, 1 gene for blue. The phenotype would be the rabbit would look black, and you wouldn't know the genotype until you bred it. But then you breed it, and in the litter, you get some black ones and some blue ones, and you say, okay, that's a heterozygous one. So that's the difference between phenotype is what you see with your eye, genotype is what you deduce from a breeding experiment.
So, Rob, have you done things other than goldfish and fish-wise, like angels, or and so the genetics, for example, of angelfish is really well known, right?
Yes.
And way more so than goldfish.
Yeah, it's more, goldfish, it's extremely complicated, again, because of the tetraploidy issue. You can make the little Mendelian Punnett square you've probably seen where you have the capital A and the small a, B for the number of genes, and come out with a number of different types. It's pretty straightforward with a standard diploid organism. Once you get tetraploid, and it's like, well, this goldfish is black, but this one is sort of black with a bronzy belly, and this one's kind of a chocolate, and this one kind of looks blue. And you're like, well, what's the discrete genotype of each of these goldfish? And it's not that simple. It may be that it's the combination of a heterozygous black that's also carrying a gene, a MAT gene. These things start interacting so that the phenotype that you see is actually a combination of the expression of multiple genes under different environmental conditions. We know sometimes bright sunlight can make goldfish turn a deep black. So there's so many factors going in, it's hard to just say, well, these goldfish have DNA for black body color, like the black ranchu. A lot of people bred those many times, and all they ever got out of them was these olive fish.
Right.
And they had to be the same genetics as the parents, but what was the difference? And
it took a lot of people in this room and a couple other people in art to figure out that when the fish are really, really, really young, that the melanophores, the black pigment bodies, proteins, am I saying that right? Melanophores are proteins.
Melanophores are the cells that produce melanin.
Okay, yes.
And melanin, interestingly enough, is a cell product that is not a protein. So the protein that's produced is called tyrosinase. It's an enzyme, and tyrosinase produces melanin as a byproduct of its action. So it looks black, but black isn't actually a direct coded protein. The enzyme is.
Well, the reason it's an interesting story because for years we've tried reproducing black ranchu in the United States, and we've now figured out how to do it. And so it's pretty cool. I mean, you can get an inky black rancher pretty easily, but it has not very little to do with genetics. It has to do with moving the melanophores to the surface when the fish are really, really young. And so, in fact, you can actually treat eggs to make the fish blacker. And we, you know, if you want to do black ranchu and you're buying them, we can give you now a formula, and the formula goes something like this. Once the eggs hatch, you raise the temperature to 80 degrees, and the melanophores start to float towards the surface of the fish. If you want really black fish, you should grow them in a black tub. You grow them in a glass tank with a glass bottom, they never get black. You need to have extremely intense light or they never get black. If you get them exposed to the bright sunlight very, very early, the melanophores not only float in the body, but they float in the eyeballs. And so that if you look at black ranchu and they're truly black, a lot of the fish look like they have no eyeballs. And it's because the melanophores are literally covering the surface of the eye, and the fish are relatively blind, or mostly blind.
And there's also, you may notice if you've ever taken a calico colored fish, if you're doing a water change, and you put it in a white basin, that it blanches out, all of a sudden its black spots sort of disappear and it looks, so there's, these melanophores have the same ability as what is in a chameleon skin, where they shrink and it's a slight form of camouflage where the fish can darken and lighten its color by expanding the melanophore to match the background. And I think it's an interesting observation that any of the black, solid black goldfish breeds are always an eye type. Gary mentioned that the boxer head Thai black ranchu, you can't even see the eyes, or they're grown over, or they're very black. Black Moors have a big eye. They did an experiment, this sounds cruel, but they actually took small goldfish and they ablated the eyes and they turned black. And so we're wondering if something to do with vision, a lack of vision, where the goldfish isn't sensing the light, is allowing the melanophores to be maximized and look really dark. Whereas if a goldfish that can see, obviously you put it in the white basin and it gets lighter. So that might be part of it.
If you take those steps, the first 2-3 years, is that pretty permanent, or does that?
No, it's not the first 2-3 years. It's the first 2 to 6 weeks.
Okay.
And so for black ranchu, you basically, once the fish are hatched and swimming, you raise the temperature to 80. You put them in black or dark blue containers. You leave the lights on 24/7. What works best is to buy an infrared reptile light if you're doing it indoors. And you basically, if you look at the literature for melanophore movement embryonically in fish, you have a very short window to get those melanophores to float near the surface. They're often, if you don't have bright lights, the reason they never got black is the melanophores, when they were able to migrate, and the fish were either embryonic or very, very, very young, they were able to migrate. As the fish develops the ability to migrate to the surface and give you black fish goes away. And so you got these olive green fish because melanophores are stuck inside the dermis, deeper layered into the fish, under the scales, and so you never get the blackness. For years we were throwing fish away, like, what are these guys doing? And we just didn't know how to raise them. And we found a couple articles, read the articles, set up my whole basement 3 years ago was nothing but a black ranchu experiment. And we tried, feeding them clay, feeding them color-enhancing foods, and all these other things. Glass tanks wrapped in black garbage bags, black tubs, and we now have a formula, and it works really well. And once the fish turn black, and you put them in a glass tank, they will fade if it's all glass or a white tank. But as soon as you put them back in a dark tank, they go black, because the melanophores are active at the surface. They've already floated.
And you may have,
it's actually migration of the melanophores towards the surface. It's not floating.
and you mentioned the 2 to 3 year time frame. I don't know, you may have seen some tricolored telescopes, which are real black on top and then have bright orange. Those are super slow demelanizers. So a normal goldfish with normal vision usually completes the total demelanization within a couple months. But the telescopes, if they still have those demelanizing genes, maybe it has something to do with the vision in the eyes, but it's really delayed, and you get these what you see panda patterns, where the migration of the demelanization pattern gives them that stripe on the back of the black. It's the last part to demelanize.
But the genetics discussion, as I told you, is well beyond most of us, okay? But you have to start somewhere to understand that when you're going to manipulate a color in goldfish, like you have a calico fish, and you want a blue pattern, well, there's not blue pigments in goldfish. It's black pattern, melanophores, at certain levels in the dermis, okay? And once you understand that, you say, well, how can I get a blue calico fish? And if you could get bristles, and I've got bristles here for Rolly that we can look at, and they're going to get bright blue, as bright as blue as they get in goldfish. And the reason they're going to get bright blue is because they have melanophores at the right layer.
So you can just actually manipulate how long you keep them dark or whatever. Is that something you could do, where how blue you can get something, or?
I don't think it's
they
figured out.
They have to have sun exposure. They have to be in a dark container. And I can tell you that if you find a calico fish that you think you want to be blue, it better have black in the tail. Because your chances of getting a blue fish, blue calico fish, in other words, the skin is blue or appears blue. If it doesn't have black in the tail, your odds of getting blueness are very, very small.
I'm not going to spend too much time on the minutia of transcription and translation, but I'll just briefly. The DNA is the blueprint that's in the nucleus, and what happens is the 2 strands of DNA loosen up, and an enzyme called RNA polymerase sets down there. And now it may set down in that non-coding region that they used to think was junk DNA, and how well it sits in there has a lot to do with that sequence. So that's the importance of that non-coding region. But it goes along and it makes a complement to the DNA strand, but it's RNA, and it substitutes a nucleotide called uracil, which is the U. So rather than ATC, you now have AU and I think it's AU and C are the templates for the RNA. And these are called transcripts, and they get kicked out of the nucleus after they're transcribed, and they're just little short things out into the nucleus where they get converted into proteins. So making DNA to RNA is called transcription, and from RNA to protein is called translation. So translation of proteins, you can see those little mRNA, it's called messenger RNA, gets kicked out into the cytoplasm of the cell, and then these strange looking things are called transfer RNAs. What they have are 3 little nucleotide sequences that bind to the messenger RNA, and they're called codons. So say this is A, T, C, might code for a certain amino acid. These are free amino acids that come from the diet, part of the food. They're in the cytoplasm of the cell. The transfer RNAs bind an amino acid, and then using this little template, they get the order correct of the sequence of the amino acids by using this key system. And if you get a mutation in the DNA that messes up that coding, it could code for a different amino acid, which would change your protein altogether, and that's how mutations sort of happen. So I'll just keep moving on. Some general definitions. We discussed difference between a genotype and a phenotype. I didn't talk about alleles. So if you think of the nucleotides in a DNA sequence going down in a particular location, okay, across both strands, that is called a locus. You can say on this chromosome, this many number of bases down, right there. What is there? And that's the locus. And if there's a mutation, the normal wild type might be an A there, but in the mutant, it might be a T. So whether you get the A or the T, those would be the two different alleles, for that particular locus.
Rob, could you address simply, when we breed goldfish, there's sibling crosses and back crosses. Can you just briefly give us a practical reason for why you do sibling crosses versus back crosses and what it means to everybody, so maybe that'll help them?
Yeah, and I do have slides on that, so
Okay,
I'll get into that one.
Sorry, I didn't mean
No problem.
circumvent you.
So a dominant allele, if you have two different alleles, the one that gets expressed, like in the example I gave with the black rabbits, we have both the blue and the black allele present, but the offspring appear black. That is the dominant, would be the black. And you can say an eye color in humans. You often hear that blue is recessive. So two dark-eyed people can have a blue-eyed child, but it's rare that two blue-eyed people have a dark-eyed child. Okay, what am I doing here? So, in your somatic cells, these are the cells of your body, it's diploid. You would have these chromosomes, and that's your genotype. During meiosis, those get split apart. Remember, the gametes only contain one of each chromosome. So then your gametes could be either for example, the big S and the big B, the big S and the little b, the little b, and so it's going to be an S and a B, but you see what I mean? You get all these different possible combinations. So each sperm or egg would carry the potential for either one or the other of the alleles. And then it depends on what the mate is as to what you get in the children. So it mixes it up. And you have Mendels Peas is the famous experiment where he crossed wrinkly green peas with smooth yellow peas and calculated the percentages of the different wrinkly yellow ones and smooth green ones, and you get all the different combinations. Linkage is when two genes of significance are on the same chromosome so that they always get inherited together. The closer they are on the chromosome, the less likely it is that they'll ever be separated, so you'll always see those things together. But there is something called crossing over during meiosis, where chromosomes, if they're the same chromosome and they get close together, sometimes they'll swap little bits of DNA toward the end, and that is how traits that are always typically inherited together can be dissociated and jump over to another chromosome, and it's another way of jumbling up genetics.
So I have a Ranchu that actually has one beginning of a bubble eye.
Mm-hmm.
I don't know how, I mean, it looks like how a bubble eye would start. So that's kind of something with that, how that would kind of start.
It's no one would really know. The bubble eye mutation obviously was noticed somewhere. Bubble eye is a dorsalist breed, so maybe they are connected.
Somewhere in there.
Someone may have, accentuated a fish that was dorsalist and had a little bit and developed the bubble eye breed.
So are the fish Chinese?
Yeah, it's a Chinese import.
Yeah, it's interesting. There's a guy on the Facebook called Reader Agnethi who's been talking about various genetic manipulations. And if you follow the Chinese goldfish keeping, prior to Mao, there were hundreds of varieties of goldfish in China, and most of them were destroyed. And so we have less varieties than we used to have, and which is unfortunate, but that's just the way it is. But it's very common to enter a non-Ranchu into the breeding program for Chinese fish. It gives them some vigor, growth-wise, on the next generation. It also, to my viewpoint, tends to prevent you from copying that line.
That's what I heard too, that they...
Yeah, and I'm not sure that it's malicious, but it basically gives you the ability to get a fish that grows faster, okay? But the side view is if you buy a bunch of Chinese Ranchu, for example, when you breed down your F1s, your first generation, you get a tremendous amount of non-Ranchu. And it takes you several generations to recover. Whereas if you buy a Japanese Ranchu from a reliable source, you breed them, you get things that look just like the parents. And so there's a tremendous amount of add-ons and by some breeders, and it prevents you from copies walking away with those genetics.
Another
easily.
good example of that is if you ever grow plants by seeds like tomatoes, it'll say Burpee hybrid this, hybrid that, and then they have the heirloom strains. So your hybrid strains they cross two different strains of tomato, and you get hybrid vigor, and that first generation, everything's great. They're all uniform. If you try to save the seeds from those and then plant them the next year, then you're getting into the F2 generation, then they're all messed up. You'll get some with maybe little itty bitty tiny tomatoes, and you're not going to get anything like the nice hybrid that you had originally. And it's reasonable that some of the goldfish farms might be doing the same thing. They're trying to sell good-looking goldfish as pets, but they don't want you necessarily having the heirloom lines that they have so that you can take them and compete with them. That's not saying they're all doing that, but it makes sense that they might.
Yeah.
Okay, copy number. So if you have a gene, codes for a protein on a chromosome, what can happen is that during the duplication, a little end of one chromosome will hop off and add itself to the other chromosome. So basically you get a repeat in the DNA. So now this new chromosome actually has that same gene repeated twice. So in the adult animal, that protein will be expressed at twice the level that it was in the other. So that is like a new mutation that can be passed on, where it's called a copy number difference, where now you're getting 2 times as much of the expression of a particular gene.
Do you have any specific examples of what that would look like, an extra long dorsal fin or like extra blue eyes or something?
Things like, it depends. If the protein, what its function is, if it is an enzyme, like I can try to make one up, so tyrosinase, that one that generates melanin, if you had extra tyrosinase, that enzyme produced because of a copy number difference, then that fish might be a darker fish, or maybe that's what does account for black moors and the really deep color. It's hard to know in goldfish because we know these principles exist mostly from the study of human genetics. No one's putting this sort of time and scientific knowledge into the actual study of goldfish breeds, so we're all hypothesizing. There's no brilliant scientists out there who have all the answers, but we're trying to apply what we know from other research to a mechanism that would also happen in goldfish. Okay, we're getting into the inbreeding, and I'm sorry for this picture, but I was Googling inbreeding, and this is the first thing that came up, so I thought I'd put it in there anyway. When it works, it's called line breeding, and when it doesn't, it's called inbreeding. And I just want to say, inbreeding everyone hates the sound of inbreeding. They always attribute all bad things to inbreeding, but you have to keep in mind all pure strains of any species are here because of inbreeding. You can't have purebred anything without inbreeding, so it is necessary. And a lot of the concerns that we have about inbreeding are related to fears of what we've seen happen in humans when closely related individuals reproduce, as well as mammals. I mean, it's more of a tragedy when your dog has two-headed puppies and such than, you know, a goldfish has two tails. You've got thousands and thousands of goldfish babies. You're planning to cull them anyway. You're actually looking for the freaks because you don't want a comet. You want, you know, a dorsalis, bubble-eyed something or other. So these are the result of inbreeding. So the downside of inbreeding in something like goldfish would be loss of vigor and loss of the ability to breed, weakened immune systems, things like that. At that point, the inbreeding is a problem for the fish. But as far as just coming up with these different phenotypes, that's actually what you're after, and it's just a matter of a disciplined selection process and using that. And there was just an example of it was popular in the royal families to marry amongst each other, and there were examples of hemophilia and different diseases and deformities that started appearing in royal families, and they kind of stopped doing that.
Yeah. Interesting.
So allele fixation. So if you think about the genome of a goldfish, at each locus, probably greater than 95% of the loci have the exact same DNA nucleotide on both chromosomes, or both strands. Okay, so they're all homozygous. But then there are mutations. These are particular loci where there is variability, where it can be either an A or a T at some percentage frequency in the population, and that accounts for the variability in the species. If you keep breeding brother to sister, generation after generation after generation, you eventually lose that variability, because sooner or later an offspring, it'll kick out the, say you imagine the big A and the little a, you're gonna have a baby that's born with two big A's. Now that little a is lost forever if you're only breeding, you know, generation after generation. Sooner or later, it's after 20 generations of breeding, you'll get to 100% homozygosity, where all of the individuals will have the exact same genotype and there's no genetic variability at all. That usually presents survival challenges for the organism, but it is possible. The lab rats and lab mice that they use in research are homozygous strains that have been bred brother to sister for many, many generations. And it wasn't easy to do. They might have started out with 50 different crosses, and a lot of them get weakened and die off, pretty early. But it's the luck of the draw. If you eventually get a strain where this particular strain has vigor, and we're at the 20th generation, then you've got a stable strain, and that's valuable for research because you can say all the individuals are genetically identical. It's like a huge twin study.
But if you do that, if you do sibling to sibling crosses, most of us are small hobbyists, and so you buy a pair of fish at Seagrass or from me or from Rob or Joshua, and you breed brother and sister, very common, right? Because you bought two fish and they're going to spawn. But that's great for 2 or 3 generations, and then you start having issues, and the issues twofold. One is that we find that there's no vigor, and that the vigor related to wanting to breed goes away, and so you'll have 50 and they're really beautiful fish at the end of the year, and the following year when you go to spawn, they just don't spawn. They're not interested in spawning. You're really not sure if you have males and females, and so we've seen that in the hobby. We've seen it especially with Philadelphia veiltails in the early 90s, where the number of breeders in the United States were 2, and we hadn't done an outcross for decades, and they were doing sibling and sibling crosses. They didn't know any better. We didn't know any better, and we ended up losing the blue veiltail specifically. But before we lost it, we had all these crazy things that looked like diseases show up in those fish, and you could keep them under control with chemistry. But they were prone to having their fins rot off. They were prone to getting globules of mucus on their tails. They were prone to develop a cart mouth instead of a minnow mouth. And all these things, when you do sibling crosses for several generations, you end up with making the gene pool homozygous and what I would call shallow very, very quickly. 4 generations, 5 generations, you're there. And so as you breed goldfish, if you're going to breed and you have a line of goldfish or you have somebody else's line of goldfish, it's important that you pay attention, and one, when the fish show up with these unusual deformities, that you don't keep them. And the other thing is that you recognize the signs that it's time to do an outcross or get some genetics. Right now, for example, Rolly's taking home some English veils, which I brought today. Well, we only have 2 sources of English veils that are historic in the United States. We've had them for several generations. And so we're actively looking for new genetic material. For example, the famous goldfish breeder Harris Zane has English veiltail genetics that are different than mine. And so I had to figure out how to get fish from Hawaii, and for example. And the Bristol Shabunkins that I've had now, I've had Bristol Shabunkins since mid-1980s, but we've imported stock 3 times. But now I'm nervous. We have really, really nice stock, way improved as compared to where we started. But we can't get Bristols from England anymore. So we found a guy who went to England He's a flutist, classical flutist, lives on the West Coast. He's got 2 genetic pools of fish that aren't part of the mainline Bristols, and so we're doing a swap this year. And so it's critical that you pay attention to that, because you work decades sometimes on fish like his Montzuamas. Rob's got this Montzuama line of ranchers, and they've been around for a long, long time, okay? Almost 20 years.
Mm-hmm.
Close.
Yeah.
And if you don't figure out how to add those genetics without ruining the line, you end up, when you're doing all this work and the fish just look at you, they're not going to be.
Well, another thing to keep in mind, just because you've had them for 20 years doesn't mean that you've crossed sibling to sibling 20 generations down.
Great point.
It's usually good to use your Oya, which is a 3-year-old female. And the reason is they throw more eggs, and their eggs are bigger, and their fry are larger when they hatch out, and the viability is better. I don't see that sort of requirement from the males. I think it's okay to use the Tosai males. The sperm is fine, and you can get the genetics of the next generation and go. If you're female, your Oya female is a really great one, and you want her to be like a cornerstone of your line. If you breed backward, it's called a back cross, from say an F2 or an F3 back to one of the really good fish from a previous generation, you're reintroducing the genetic diversity that may have been lost in the subsequent generation. So say 5% of that Oya female's loci have some variability in them. If you take a fish way down here, it's lost some variability, so only 2% variability at its loci, and then spawn it back to that female, it's not going to be less than 2. You're actually going to be somewhere between the 2 and the 5. You see what I mean? So you can keep the right genetics in the pool without giving up the variability. You want to try to have homozygosity at the loci that account for the traits that you're breeding for, and you do that just through selection. Say, I always want to have a square head, I always want to have a dorsalis back, small scales, things like that. But you want to maintain genetic diversity at all the loci that say govern the immune system and its ability to produce a wide range of antibody types and things like that. And that's how you keep your fish healthier. You also have to know where you're starting from. Gary mentioned, 2 to 4 generation sibling crosses, and you're there. If you do a cold outcross, even between different breeds, I think you may have 10 generations before you're there. But a lot of times when you receive fish from a breeder that are an established strain, they are pretty there already. So you have to be careful. And a way to avoid that is you can start your A strain and your B strain. So say you get a group of fish from a breeder, you do a couple different pairings, and then you separate those, and you can do sibling crosses for a few generations, and then take a male from this strain and swap it with the female from the other strain and reintroduce some of the vigor that was lost, because they're not going to lose the exact same diversity at the exact same loci if you have 2 different strains.
And there's some tricks that are pretty simple that you can play to prevent inbreeding. And I'm going to cite you a couple that the group of us are doing. For veil tails, there's only 6 breeders in the United States that are routine breeders, not half-stance breeders or semi-series. So we switch young every 2 years at least, sometimes more. And then the guys that are local to me that can drive to my house, we trade adults. And so, they'll come to my house and they'll bring 6 or 8 fish, and I'll take some of them and they'll take some of mine. And so we're changing the genetics. And that's kind of important. Last year, I bought this strain of Japanese Ranchu called Hayashi, and people really like them. They were pretty expensive. They were imported from Japan to Genki Koi in San Jose. Well, I bought 10 Tosai, which are 1-year-olds, and I brought 4 Nisai, which are 2-year-olds, different parents. And so I brought different age classes in, and by bringing different age classes in, I got different genetics. So I'm planning for different genetics. This year, my Bristols are pretty exceptional, I think, shape-wise, at least Joshua thinks. So he's been to my house, Rob's been to my house several times. And what I did is I drained my koi pond and I got some 6-year-old fish. And I bred my 6-year-old fish to my 1-year-old fish, back cross. And we really got some nice vigorous fish this year. And we think we've improved, once again, the genetics. So if you would go to my fish room, there's electrical tape. I love that colored vinyl electrical tape, you know, and so there's notes on all of them. You can see the dates and what the crosses were, and if they have BC on it means back cross. And so, you know, the back crosses are important. If you're starting a line of any fish, and you're buying a line of fish at Seagrass, or be cognizant of the fact that if you're going to make that into a line and you're just not keeping them for fun, that you should look really hard and see if there's multiple sizes of what appear to be the same fish. Consider buying a bunch of smaller fish because they're affordable, and buying 1 or 2 older fish, which probably have slightly different genetics.
Another thing, as you're trying to pair up fish, breeding to a standard, trying to reach a specific goal you have in your mind, you always want to use somewhat related fish. Like with the Ranchu, you'll see all these initials, like mine are Matsuyama Oishi, so it's M-O, and then someone crossed these to the OKS, which is, what's that, Oishi?
Oishi Kajiyama times Suzuki.
And so those are 3 other lines, and they were working with those for a while, and then we take some of those and cross them back to mine. So they share the same genetics, but they're cousins, and they've kind of been working in the same family. Way back when, I had some Chinese Ranchu, which have a different body type. They're not the long top view fish. And at that time, they were just the fish I had, and I tried spawning them, and I did. And I crossed a Japanese Ranchu, because I got all males at that time, with a female Chinese Ranchu, and the fry all had dorsal fins. So just because 2 fish have the same phenotype, dorsalis, does not mean they have the same genotype. There's many different paths to get to the same phenotype. They may have completely different mutations at different loci that both cause the dorsalis, and when you cross them with unrelated fish, all you're doing is recovering the wild type, which is a normal dorsal. So, you know, you can't assume that a veil tail in one breed is governed by the exact same gene as it is in, say, a veil tail. They might be a little bit different, and if you cross them, you may get a long-tailed fish, but you may not get the same results that you're thinking. For example, my Azumas have long tails, but they're all deeply forked. A comet goldfish has a deeply forked long tail, but broad tails and veil tails have a straight edge. So it's something different going on with different genes governing how those long fins are developed.
And if you're breeding fish, siblings, like most of us get stuck doing at one point in time, and you decide you want to improve the stock, you can often buy fish. If you bought it from an importer, and you want to improve the stock that you have, and it came from, say, Fish Corner, you know, with the Gun Chusakyo, or it came from Danny Aranda's, Ken Fisher, you can pretty much find out if the fish they're selling today came from the same farm as 2 years ago by just asking him. So, you know, Gun has he's getting the vast majority of his fish from his family farms. You know, Ken Fisher has been using the same importers for decades in some cases. And so if you go back, you have some fish, you want to add an outcross because you want bigger, you want a better color pattern, or you want to put speckles in the calico fish, I suggest you go back to where you got the fish, the importer, because if you ask them, they might not know, but chances are they're still getting them from the same farm. So it helps you keep the genetics similar.
Okay, so pedigrees, just so you know, the definition of line breeding is having the same individual show up on both sides of the pedigree, and it's done extensively in thoroughbred racehorses. And if you look, here's an example. You may recognize some of those horses' names, but Northern Dancer, all the way over one to, you know, the last generation over here, appears in both the dam and the sire's pedigree. So that's an example of line breeding. It's not too extensive, but and even before then, you might have others horses that are showing up on both sides. Again, when we talk about back crossing, one of the projects that I was working on, unfortunately I lost a lot of these fish. I don't know if you're familiar with Amy Sim Shanka, has these beautiful broad-tailed black moors. A few of us were interested in creating a calico version of this. So I had Azuma nishiki, which are a calico fish. I crossed those to the broad-tailed black moor. And in the first generation, I did get some very nice normal-eyed Sakura looking fish, and kept breeding those back to the black moor, selecting for the ones that have the calico coloration. And you can do that, and what you're doing there is you're preserving that locus for the calico coloration while trying to recapture all the loci that were in the black moor, I mean the body type, the finnage, the eye type. And here's an example where you have two different chickens, and you cross them, and then you keep back crossing to that one male, but selecting for the one with the more white on the body. So you can eventually get a very similar looking bird, the only difference being that white body, but being 86–90% the same genetics as that original parent after several generations of back crossing. And that's not inbreeding, because remember, you're going back to the same individual, so you're not going to have any loss of genetic diversity. You're just going to make it more and more similar to that original bird. Now, these are part of that cross that I was telling you about, where I crossed the black moor to the Azuma. I did this talk, those were early examples of some of the variety that I got out of that cross. And some of them turned into pretty nice fish, and then the eyes started coming out in like the third generation. Okay, I won't go into all this. These are some of the Japanese studies that were done in the 70s that I can send you the reference if you wanted to, where he looked at genotypes and color mutants in goldfish and kind of figured out the whole calico coloration with transparent scales by doing different types of back crosses. He also looked at albinos and black individuals. And that's the end of this slideshow.